Greetings everyone, myself Sugandha Sharma. My topic for presentation is Scanning Electron Microscopy. In this presentation, I will be discussing about the introduction and brief history of the SEM, what happens when the electron beam falls onto the sample, how it interacts with the sample, what are the working principles of SEM, what are the conditions required for SEM, and the instrumentation of the SEM, how the formation of bright and dark spot of SEM images takes place, what are the important aspects to be considered in SEM, the detectors which are used in SEM, and also the positive aspects and the limitations of the scanning electron microscopy. So to begin with, what is electron microscopy? The electron microscopy is just like the optical microscopy, but the difference is that in electron microscopy, we use the electrons instead of photons to image the sample and gather the topographical information. In electron microscopy, the microscope uses the high energy electron beam as its characteristic to magnify the image of the object. SEM, which stands for scanning electron microscopy, uses a beam of high energy electron to focus and scan the surface of the sample. The electrons interact with the sample that produces image by scanning the sample surface and focus electron beam. First scanning electron microscope was developed by Manfred von Andren in 1937, and then in 1940s Ruska developed SEM, which uses electron magnetic lenses to focus the scanning electron beam onto the target surface, and then scattered electron contain the information about topography and the structure of the sample. In 1965, first commercial SEM was developed by Cambridge Instruments. Now, as it can be seen from this picture that when the incident beam is allowed to fall on the sample surface, various types of interaction occur which leads to the generation of Auger electrons, secondary electrons, backscattered electrons, X-rays and cathodoluminescence. And some of this incident beam may get transmitted from the sample and give rise to inelastically scattered electron, unscattered electron and elastically scattered electron. All these transmitted electrons are studied from transmission techniques, which is transmission electron microscopy, TEM, and these reflected generations from the interaction between incident electron beam and the sample are studied under SEM, that is the scanning electron microscopy. It can be seen from this figure that as the primary electron beam falls onto the sample surface, it gives rise to this green part, this some onion shaped like interaction inside the sample, which is known as interaction onion, which gives rise to different types of electrons and different types of scatterings in the from the sample. As the primary electron beam interacts with the sample and penetrates to the depth of about 10 to 30 angstrom, it gives rise to the Auger electrons, which gives us the surface atomic composition of the sample. Next, as it moves deeper, as the primary electron beam penetrates to the depth of approximately 100 angstrom, it gives rise to the secondary electrons, which gives us the information about the topography of the sample, like if the how is the sample surface, like if it's porous or it is smooth. All this information is gathered from the secondary electrons. Now, next, as the electron beam penetrates to the depth of less than 1 to 2 microns, it gives rise to the backscattered electron, which gives information about the atomic number and the phase difference of the sample. As the primary electron beam penetrates more deeper, that is to the depth of approximately 1 to 3 microns, it gives rise to the thickness and the atomic composition of the it gives rise to the X-rays, which give us the information about the thickness and the atomic composition of the sample. Next, let's come to the working principle on how does the SEM work. In SEM, the electron gun, which is located at the top of the instrument, acts as a source of electron, which generates the high energy electron, and the electron beam is then forced to travel vertically down the SEM column and is focused onto the sample with the help of magnetic lens, which helps this electron beam to travel parallel to each other, like it is because of this magnetic lens that the electron beam travels in a straight line down the SEM column and falls directly onto the specimen. And the scanning coil applied here around it is applied around the objective lens. It helps to it helps to make sure that the electron beam falls onto the top of the specimen in a particular periodic interval. Next, let's come to the sample preparation. As we gear up to get the SEM image of any of our sample, some of the samples are such that it requires some pre-treatment. So the pre-treatment means we require to prepare the sample so that we can get the best possible SEM image of our sample. So here I have listed few techniques of sample preparation, which includes mounting. Mounting means just keeping the sample onto the stub. Stub is the main base on which the sample is kept, and then stub is placed onto the sample stage. So for powdered samples, we use the carbon tape for placing our powdered sample onto the stub. For bulk samples, we fix the sample onto the stub with the help of conductive glue. And for wired samples, we use the stable fixation using the conductive tapes. But one condition is that we should avoid any of the air traps inside the bunch. Next is we need to do the conductive coating so as to avoid the charge buildup on the sample surface, which may arise due to the fall of the electron beam onto the sample. Next is drying. We need to dry the sample completely so that no moisture is left into the sample, because if any of the moisture is left into the sample, it may lead to the destruction of our SEM instrument. Next is embedding. Embedding means that the sample kept is embedded in some stable resin. For example, we can use epoxy resin to just embed our sample into it. Next is polishing of the sample. Polishing of the sample is to be done so that we can get the good quality of backscattered electrons. Next methods include etching, freeze fracture, cross-section cleave. All these methods help in sample preparation. Now, there are two methods which are used for producing a thin layer of metal ions or metals on the sample surface, which include the sputtering and the evaporation. Sputtering means bombardment of sample with the ionized gas, which knocks the metal atoms off the surface of the target and let them settle onto the surface. Next is evaporation. Heat metal or carbon in vacuum chamber until it evaporates and settles on the sample surface. Next, let's have a look on the SEM column. The SEM column includes the electron gun, anode, condenser lens, objective lens, the sample chamber. However, this electron gun, condenser lens, objective lens and anode are kept inside the vacuum chamber, and the sample chamber is kept outside the vacuum. The SEM column is kept inside the vacuum because if any of the air is present inside the SEM column, then the electron beam which is being generated from the electron gun, the electron beam which is being generated may bombard with the gases and to ionize the gases so it will lose its energy before falling onto the sample. So to avoid this, the SEM column is kept in the vacuum. Also, the vacuum helps the electron beam to stray away from its path. So as the electron beam may travel straight in onto the sample. And the sample chamber is not kept in the high vacuum because some of the samples may not withstand the high vacuum. So to maintain that, the whole instrument, the SEM column is kept in high vacuum. However, the sample chamber is not kept in the vacuum. Next is the accelerating voltage. The accelerating voltage which arises due to the potential difference between the electron gun and the anode. For different materials, different accelerating voltage is required. As the higher accelerating voltage is applied, greater will be the depth of penetration of the electron beam into the sample, and hence more interactions will be produced. For dense samples, we generally need high voltage because for dense samples, it will be difficult for electron beam with low voltage to penetrate into the sample, and so since the sample is dense, it will make less informative for us to study it. So for this dense sample, we need high voltage so that it can penetrate deeper into the sample and can generate a bigger interaction onion in the sample, and we may get and we can get as much possible information from the sample. Next, let's come to the instrumentation part of the SEM. The instruments of the SEM include the electron gun, which acts as the source of electron beam. The electrons, which are discharged from the filament, move down this walnut cap, which has the negative potential applied to it. The filament, it acts as the electron source. As this filament is applied the electric current, it generates the electron, and here the charge gets built up, and this walnut cup has negative potential applied to it, so it keeps electron away from it. And as the charge is developed inside this walnut cup, the anode plate, which is positively charged, which has positive potential on it, pulls the electron towards it and acts as the moving moving force for the electrons to travel down the SEM column. And the types of filaments that can be used in electron guns include the tungsten filament, which is hairpin shaped, the thermionic type filament, which has LaB6 in it, which is much better than tungsten filament but it is much more expensive than tungsten filament. Field emission type filament also uses the sharp tip of tungsten filament, but it gives more brighter image as compared to regular tungsten filaments. Next, let's come to the electromagnetic lens. Electromagnetic lenses avoid the straying of the electrons in the different directions because the electromagnetic lens has magnetic field present inside around it. When the magnetic field is present around it, it forces the electron to pass down the SEM column onto the central axis and avoid the straying of the electron in different directions. Also, the velocity of the movement of electron depends upon the amount of field applied. As more electric magnetic field is applied, the velocity of the electron, that is the movement of the electron, will speed up with as the magnetic field is increased around the electromagnetic lenses. Next, let's come to the objective lens system. The objective lens system has electromagnetic coil outside it, which controls the amount of magnetic field applied. The objective lens system consists of the scanning coil, the stigmator, the beam limiting aperture. The scanning coil scans or sweep the beam in a regular fashion onto the sample. The stigmator is used to correct the astigmatism if present. Next is beam limiting aperture. The beam limiting aperture controls the width of the electron beam that goes down the axis onto the sample. The astigmatism. Now let's come to the astigmatism. What is astigmatism? Astigmatism is the uneven focus of the electron probe onto the sample, which may result in the distorted or blurred image of the sample. The astigmatism may rise due to the out of focus of the sample image, which may give rise to blurred image of the samples. If the current is too strong or too weak, then the focus of the sample image may not occur at correct distance. So it may give rise to the astigmatism. So the stigmator present here functions to correct this astigmatism if possible in the SEM. Let's come to the SEM images. As it can be seen from this image, this is an example of SEM image, that so many dark and bright spots are available on this SEM image, which arise due to the different number of secondary electrons being ejected from different areas of the sample when the electron beam falls onto the sample. So when the electron beam falls onto the sample surface, the sample may have some of this, like this raised area, some this valley type area which is deeper into it. So when the electron beam interacts with this sample, different number of secondary electrons are ejected, which are responsible for this contrast in the SEM image. As soon as the electron beam falls onto the raised area of the sample, it gives rise to more possible secondary electrons. It gives rise to the more possible secondary electrons, secondary electrons in the term that more space is available around the secondary electrons, which may give rise to the maximum possible number of secondary electrons to get detected. So it gives the bright spot of images because so many secondary electrons are being ejected. So many secondary electrons will be detected by the detector, and hence it will give us the bright image. However, if we consider this valley area, when the electron beam falls onto it, it is not possible for this area to eject as many secondary electrons as this raised area, because from this area, when the electron beam falls onto the sample, the secondary electrons which are ejected will have to cross this so many hurdles, we can say hurdles, to reach the detector, which will make it less possible, or it will make the lesser in number of secondary electrons which will be detected, and hence it will give the dark spot. So this is how the bright and dark image contrast of the SEM images is made. All this is termed as the edge effect. The edge effect refers to the discrepancy in the image or resolution or the signal intensity between the sample center and its edges. Like this edge, and this is the sample center. From the raised area we get the bright image because so many secondary electrons are being detected from the detector, and from this valley area we get dark image because not many secondary electrons are able to reach the secondary electron detector. The important aspects of the SEM include the magnification. The magnification is the ratio of image length to the scan length. Smaller scan areas for the same size of the image result in the higher magnification. Like if we are scanning a image and we focus or scan the small portion, it will result in higher magnification. That is, we will get good higher magnification, more amount of magnified image of the small portion. Resolution is the ability to resolve two closely spaced points, like it is the ability with which we can see two closely spaced points to distinctly. Resolution in the SEM is determined by the size of the region from which the signal is being produced. For same region of excitation, the resolution from secondary electron, backscatter electron, and X-rays differ. The depth of field is the height over which the sample can be clearly focused. The depth of field is inversely related to the resolution, because which means that if the depth of field is increased, the resolution decreases. The working distance is the distance between bottom of the SEM column and the top of the sample. The working distance is directly related with the depth of field. That is, as the depth of field is increased, the working distance also increases. But as the working distance is increased, the resolution is decreased. That is, working distance has a reciprocal relation with the resolution. Next is effect of probe diameter. Probe diameter is the diameter of the incident electron beam which is focused onto the sample. As the diameter of the focused electron beam is increased, we get a bigger spot size onto the sample, which will result in the bad resolution because the electron beam is focusing onto the large portion of the sample. However, if the probe diameter of the electron beam is decreased to the small spot size, we'll get good resolution because the electron beam is focusing onto the small portion and giving us good resolution of that area. Next is effect of probe current. As the probe current is increased, the probe diameter is increased, and hence as probe current is increased, the probe diameter is increased, and hence we get low resolution. See, as it can be seen from here that if more probe current is applied onto the sample, there will occur more damage onto the sample surface, and hence we'll get deteriorated resolution of the sample image. And as the lesser amount of probe current is applied it will damage the sample lesser, and hence it will provide us with good improved resolution. Next is the effect of oscillating voltage. The oscillating voltage is the voltage difference between the filament and the anode, which oscillates the electron beam towards the SEM column. Is when the electron beam is forced to fall onto the sample surface. If then the two conditions applied. If the atomic number of the material is low, and if the low voltage electron beam is allowed to fall, then it may interact considerably, like it may interact moderately with the sample. However, with the sample with low atomic number is made to interact with the electron beam of higher voltage. Then the higher voltage may penetrate much deeper into the sample and hence will give much more information about the sample. However, on the other hand, if the material with high atomic number is used and the voltage of the electron beam is low, then it may not give us much information about the sample, because low voltage will make it less possible for electron beam to penetrate deeper into the sample and hence will give us less information. However, if the electron beam of high voltage is used and is penetrated into the sample, then we may get the good information, moderately good information about the sample with high atomic numbers. So for materials with high atomic numbers, high voltage should be used. Next, we'll come to the detectors which are used in the scanning electron microscopy. The backscattered electrons are detected using the backscattered electron detector. It detects the elastically scattered electrons. The backscattered electrons are placed right above the sample stage. It has a four-quadrant backscattered electron and it is applied in an annular form around the optical axis above the sample, which allows maximum number of possible backscattered electrons to get detected by it. Here it is placed just right above the sample. So this is the sample stage and this is the backscatter electron which is placed right above the sample stage. So maximum number of electrons are detected from the backscatter electron, and the backscatter electron uses the four quadrant solid state backscatter electron detector, in which each quadrant gathers the electron and gives out the information about the sample. Secondary electron detector detects the inelastically scattered electron. It provides the topographical information with best possible resolution. In the secondary electrons, when ejected from the sample, the secondary electron detector is placed at some angle from the sample stage. As the secondary electrons are ejected from the sample space, the secondary electron detector has positive potential applied on the top of it, which attracts the secondary electron towards itself, and the secondary electron detector has the scintillator in it, which converts the electrons into the light photons. The photons are then the photons signal is then amplified using the photomultiplier tube and to give the signal onto the sample. Now, the positive aspects of the SEM microscopy. It provides us high resolution. It gives us high depth of focus about the sample. Low mass of the sample gives us the higher resolution, helps us get the high resolution of our sample. Having multiple detectors, since in SEM, as we have seen that there are multiple type of detectors which are applied in the SEM microscopy, it makes SEM a versatile tool which can be used to gather various informations about the sample, and also SEM provides valuable solutions for many different applications. Also, SEM has very few limitations also, which includes that SEM instrument is extremely expensive. Its operation required environment free from vibrations and also free from electromagnetic elements. The consistent voltage and cooling system are must for the smooth functioning of the SEM. The time consuming sample preparation is also required because this, if sample has moisture in it, then the instrument may collapse. Also, the radiation safety concerns are included in the SEM. Thank you.